What makes you so different from your siblings or friends? Your eye color, personality, height? It's actually mostly the proteins you possess. They control everything going on inside your body right now, from most of your hormones to your enzymes. So how is this great diversity in proteins introduced from very few genes? The answer lies within two big words: post-transcriptional modifications. It all starts with the process of making proteins in your cell, called protein synthesis. The regions of your DNA, called genes, such as this one, are transcribed. Genes are made of DNA nucleotides: adenine, thymine, guanine, and cytosine. In transcription, the gene unzips, and an enzyme called RNA polymerase adds and forms bonds between RNA nucleotides, molecules similar to what DNA is made of, making a long molecule called pre-mRNA. And now, to what you've all been waiting for: post-transcriptional modifications! mRNA has a lot of misleading parts that don't code for the protein, called introns, while the parts that actually code for the protein are called exons. For the mRNA to be useful in making proteins, the introns need to be removed. And luckily, our cells have an enzyme for that called a spliceosome, which splices introns out and joins the exons together. Sometimes, some exons are also spliced out to make a completely different final mRNA and a completely different final protein. This is known as alternative splicing. So one mRNA can form so many different proteins. But wait, hold on, we still haven't made a protein yet. We still only have an mRNA strand. So how come the mRNA isn't destroyed by all the evil enzymes in the cytoplasm when it exits the nucleus? And how does it even exit the nucleus in the first place? Well, there's still two more processes in post-transcriptional modifications. I didn't mention this earlier, but when RNA polymerase starts making mRNA, it adds 3 phosphate groups that act as a starting point to add nucleotides. In capping, the last phosphate group has to be replaced by a guanine nucleotide, forming a cap. Polyadenylation occurs on the other end of the mRNA, where an enzyme called poly-A polymerase adds hundreds of adenine RNA nucleotides, forming a poly-A tail, which protects the mRNA from those enzymes I mentioned earlier. Now you have a mature mRNA strand that can attach to special proteins which take it to the ribosome, where the mRNA is translated, forming a protein chain. Post-transcriptional modification is one of the many processes that make you unique. My name is Jennifer, and thank you for listening.